Helicase is the enzyme that opens up the DNA molecule and allows for the DNA to be synthesized and replicated. Single-stranded binding proteins, or SSBPs, are the proteins that go on the outside of the newly opened DNA molecule. RNA primase is the enzyme that comes to the strand where the open side is a 5 and the closed side is a 3, and lays down primers that allows for the DNA to be replicated.
DNA polymerase III is an enzyme that attaches to the primer laid down by the primase and adds bases according to the base pairing rules, complementing each existing base. The DNA polymerase III can also backtrack on its mistakes and edit them to be correct. Errors typically occur 1 in a billion. DNA polymerase I is an enzyme that removes certain areas of primer and Okazaki fragments and fills it in with the corresponding DNA molecule. Since the DNA polymerase I is not attached, the ligase finishes the job of the DNA polymerase I by connecting the segments to the rest of the DNA strand. After ligase performs its function, the DNA backbone is filled in and no more Okazaki fragments remain. Topoisomerase is an enzyme that loosens the DNA strand before it is separated. The tension from helicase makes the double helix very tight, and topoisomerase counters that. It also reads the DNA strand from 3' to 5' and adds bases from 5' to 3', creating a lagging and a leading strand.
Now, the lagging strand is ridiculous, because instead of being like the leading strand, where it goes in a linear fashion, in a straight line, and makes it easy for honors biology students, instead it has to be really complicated and difficult. So what happens is that since DNA has to copy from a 5 to a 3 direction, it can't do that on one of the DNA sides. So what it has to do is lay down primers and chase itself as the DNA molecule opens up further and further.
The 5' and 3' ends of DNA. The 5' represents the terminal phosphate group, and the 3' represents the terminal hydroxyl group. Because DNA is antiparallel, the new strand is formed from the 5' to the 3' ends. DNA synthesis always happens from the 5' to 3' ends. The 5' and 3' are the locations of the carbons of the sugars, and they are located on the 4 ends of a DNA strand. Okazaki fragments are small fragments of DNA that is synthesized on the lagging strand during DNA replication in the 5' to 3' direction. The leading strand is a single DNA strand replicated in the 5' to 3' direction during DNA replication, and unlike the lagging strand, it is synthesized continuously.